Hello everyone, today we are going to discuss about exogenous antigen processing and presentation. Antigen presenting cells. Antigen presenting cells are a heterogeneous group of cells that include macrophages, B cells, dendritic cells, etc. They interact with T cells to link innate and adaptive immune responses. A wide spectrum of cells can present antigen depending on how and where the antigen is dealt with by cells of the immune system. So, in a lymph node organ, there are three types of APCs major, and they are dendritic cells, macrophages, and B cells. So, these are the images of the antigen presenting cells. And let us see one by one. Dendritic cells, they are crucial for initiating responses, and they are found in abundance in the T cell areas of lymph nodes and spleen. They are the most effective cells for the initial activation of naive T cells. They pick up antigens in peripheral tissues and then migrate to the lymph nodes. They express high levels of addition and co-stimulatory molecules as well as MHC class II molecules which interact with the T cell receptors and CD4 on T helper cells. So, dendritic cells are able to present internalized antigens to T cells through MHC class I as well as class II molecules in a phenomenon called cross-presentation. This results in activation of cytotoxic T lymphocytes which are then available for killing of infected cells. Macrophages. Macrophages and B cells express appropriate co-stimulatory molecules for activation of immature T cells only upon infection. So the macrophages ingest the microbes by phagocytosis and particulate antigens and digest them in phagosomes, which are small vesicles formed inside it, and they present the fragments at the cell surface on MHC molecules. Then B cells. These B cells bind to specific antigen through surface immunoglobulin M or immunoglobulin D, and they internalize it and degrade it into peptides which associate with class II MHC molecules. If the antigen concentration is very low, then these B cells with high affinity antigen receptors, that is IgM or IgD, since they are in a pentameric structure and IgM, and it has high affinity in the most effective APC because other APC simply cannot capture enough antigen. So these are the antigen presenting cells and how they present the antigens. The antigen processing steps, the antigens are partially degraded into peptides before binding to MHC molecules. So the processing of antigens to generate peptides that can bind MHC class II molecules occurs in intracellular organelles. Phagosomes containing endocytosed proteins fuse with lysosomes where a number of proteases are involved. So protease enzymes which break down proteins are involved in breaking down the protein into smaller fragments. So the proteases include cathepsin B and D, and acidic thiol reductase, gamma interferon inducible thiol reductase which acts on disulfide bonded proteins. Alkaline agents such as chloroquine or ammonium chloride diminish the activity of proteases in the phagolysosomes and therefore interfere with antigen processing. In laboratory studies, the requirement for internal degradation of antigen by APCs can be circumvented by the use of synthetic peptides. Antigen processing. So an antigen is internalized and it is partially degraded in phagolysosome and it is presented on the MHC molecule. Exogenous antigen presentation. The extracellular proteins are internalized by phagocytosis or endocytosis. Endocytic processing pathway takes antigen to several compartments with different lowering pH which activates the hydrolytic enzymes. So first is early endosome, which is the pH between 6 to 6.5, and then late endolysosome, pH 4.5 to 5.0, then lysosome, which is pH 4.5. So antigen presenting cells has unique compartment called MIIC, where the final degradation and protein loading occurs. Phagosomes and lysosomes activate specific granules that lowers the pH and kills few antigen. MHC class II molecules are loaded with exogenous peptides. The MHC class II has open groove at both ends, and these sometimes binds with an antigenic peptides designed for MHC class I molecule. So this is prevented by an invariant chain which binds to the binding groove of MHC class II molecule. The invariant chain directs the MHC class II from the rough endoplasmic reticular trans-Golgi network to the endocytic processing pathway. In the rough endoplasmic reticulum, the unfold MHC class II along with invariant chain transported to the acidified endocytic vesicle through trans-Golgi network. This cleaves the invariant chain into small fragments. So the invariant chain is digested into small fragments called clip, which is class II associated invariant chain peptide, and now the clip binds to the binding groove of MHC class II. Thus the antigenic peptides are unable to bind to the clip MHC class II complex. Another class II molecule structure is called HLA-DM, which binds to the MHC class II clip complex, catalyzing the release of clip and binding of other antigenic peptide. So you can see this in image. The invariant chain is broken down into clip, and then once the peptide has bound, the class II peptide complex is transported to the plasma membrane, and where the pH is neutral. The complex is compact and stable. It is difficult to replace with another peptide and physiological conditions. These are about the antigen exogenous processing and presentation. Thank you.